Professor Hideki Nakamura. He is the chairman and professor, Department of Hematology and Rheumatology at Nihon University School of Medicine since 2021. He graduated from Nagasaki University School of Medicine in 1992, pursued a doctoral course at the same university in 1995, and further gained experience in rheumatology, immunology, and allergy at Brigham and Women's Hospital, Harvard Medical School in 2001. He is the recipient of multiple research grants, and he is the director of Society of Sjogren Syndrome, Japan. He is a member of Sjogren's Big Data Consortium and serves on the editorial board of Sjogren Syndrome Annual Supplement, Clinical and Experimental Rheumatology. Main research themes include different aspects of Sjogren syndrome, including the clinical features, autoantibodies, role of toll-like receptors, and mechanism of salivary gland destruction. However, the topic closest to his heart is the role of HTLV1 in pathogens of Sjogren syndrome, and that is the topic for today. Please join me in extending a warm welcome to Professor Nakamura.
In Sjogren syndrome was significantly high compared to 20,000 blood donors. In addition, HTLV1-associated myelopathy, HAM, highly complicated with HAM. In addition, low prevalence of anti-low SSA antibody in HAM-SS was found compared to anti-HTLV1 seronegative SS, as you can see. Then I focus on expression of HTLV1 in SS salivary gland tissue. HTLV1 has TAX and HBZ genes, and HBZ has inhibitory function. TAX derived from PX region induces TAX proteins and also activates NF-kappa B. In contrast, minus strand-derived HBZ inhibit activation of NF-kappa B and also induces FOXP3. And the question is, what about their expression in SS salivary glands? So I used this issue in situ hybridization probes for TAX and HBZ for the HTLV1 infected cell lines, including HCT5, that is from HAM-CSF, and ATL cell lines. By using this probe, HCT5 expresses high TAX and subsequent HBZ expression. This was confirmed by real-time PCR, as you can see. The next slide is HBZ and the TAX expression in anti-HTLV1 antibody seropositive SS salivary glands. The left panel shows ATL-SS patients. In these patients, HBZ in blue signal was dominant. In contrast, HAM-SS patients has TAX in red, signal dominant expression was shown. And interestingly, FOXP3 was expressed in HAM-SS patients as well as ATL-SS patients compared to HTLV1 carrier SS or HTLV1 seronegative SS. This slide shows FOXP3 positive Treg increases in HAM-SS as well as ATLSS. So I think FOXP3 Treg might induce inflammation in HAM-SS. Other HTV infectious disease, including ATL, it is shown HBG can induce FOXP3 and subsequent inflammation was induced by stimulation with interferon gamma. The next question is transmission of HTLV1 to salivary gland epithelial cells. There are some theories to show the cell-to-cell contact between HTV1 infected lymphocytes. So we focus on biofilm theory among them. Left panel shows SCT5. This cell lines from CSF, and on the surface of SCT5, there are many 100 nanometer HTV1 virions by electron microscopy. And the right panel shows 1 hour co-culture of SGE seeds with HCT5 at the junction of SGE seeds from SS patients, and the HCT5 shows delivery of HTV1 virions, as you can see. The next slide shows biofilm on the surface of HCT5 that is derived from CSF. On the surface, there are galectin-3, agrin, or tethering shown, and also HTLV1 gag proteins are detected. And they are matched on the surface of HCT5 compared to MOLT4. This is an infected cell line. After 1 hour co-culture of SGE seeds from SS patients with HCT5, we can see transmission of HTLV1 gag protein, as you can see, and also, interestingly, agrin and tethering are merged as a yellow signal. However, galectin-3 was not merged with gag protein compared to the control experiment with MOLT4 with SGE seeds. This is a hypothetical scheme of transmission of HTLV1. HTLV1 can transmit with biofilm structure, including agrin or tethering, to the SS salivary gland epithelial cells. And the next question is HTLV1 infection and induction of inflammation. What we are able to prove is that HTLV1 virions can transmit to SGECs by using biofilms, including tethering or agrin. And the next question is, can HTLV1 infect salivary gland and promote inflammation that is observed in SS? This slide shows long-time co-culture of SGECs with HCT5 that shows infection to the SGECs derived from SS patients. The green signal shows HTLV1 gag proteins, and the red signal shows cytokeratin signal for the SGECs. And after 70 hours, we can see the yellow merged view signals as an infection and integration to the SGECs. In addition, time course analysis of cytokine array in co-cultured supernatant of SGECs and HCT5 shows a time-dependent increase of cell adhesion molecules, including sorbo-ICAM1 and chemotactic factors, including RANTES, and pro-inflammatory cytokines, including IP10, compared to the control experiment with jacket. This is a quantification of molecules upregulated by cytokine array. So ICAM1, RANTES, or IP10 were time-dependently upregulated. And interestingly, we confirmed that cytokine production from HTLV1 infected salivary gland epithelial cells compared to the right panels, SGECs with Jagat-controlled experiment. SGECs ACT5 showed matched view of HTLV GAG proteins and pro-inflammatory cytokines and chemokines at 96 hours. Finally, I show the suppression of BAF expression in follicular dendritic cells. This slide shows the role of ectopic germinal center and BAF for autoantibody production in Sjogren syndrome. Around germinal center, B-cell area, there are some follicular dendritic cells, and follicular dendritic cells produce CXCL13 for B-cell homing and BAF for B-cell differentiation and subsequent selection of high affinity B cells and plasma cell production and autoantibody production by the help of follicular helper T cells. The importance of CXCL13 and BAF were already evidenced by the knockout mouse experiments. This slide shows the reduced germinal center formation in salivary glands of HAM-SS patients. In HTLV1 seropositive SS patients, the ectopic germinal center positive rate was significantly low compared to HTLV1 seronegative SS patients, and 9 HAM-SS patients had no germinal center formation, and HAM-SS patients had no CXCL13 expression compared to right panel of HTLV1 seronegative SS patients. To determine these results, we establish follicular dendritic cells like cells from human tonsil. These FDC cells showed cell surface markers that were consistent with previous reports. This slide shows HTLV1 repressed FDC cytoplasmic FDC and CXC13. The red words show the HCT5 that was from HAM CSF, and MT2 was from ATL, adult T-cell leukemia. By addition of these cells, the BAF and the CXCL13 expression was significantly reduced. And this slide shows the effect of HTLV1 infection on culture supernatant and serum BAF level. Upper panel shows BAF expression in culture medium. When I add interferon gamma, the BAF expression was increased. However, we added HCT5 or MT2. The increased BAF expression was significantly inhibited. And also, serum BAF levels was slightly low in HTLV1 carrier SS patients. This slide shows characteristics of HTV1 compared to EB virus in Sjogren syndrome. EB virus promotes autoantibody production. In contrast, HTLV1 infect SS salivary glands and induce inflammatory cytokines and chemokines, and also inhibited BAF expression and probably autoantibody production by inhibiting FDCs. This is the summary. HAM is frequently complicated with SS, but the incidence of germinal center formation and autoantibody production are low. In HTLV1 seropositive SS salivary glands, TAX HBZ also expressed in glandular tissue, and TAX was dominant in HAM SS. HTLV1 infected SS salivary gland by a biofilm, including agrin or tethering, and induced molecules involved in inflammation and migration. Finally, HTLV1 infection suppressed BAF production from follicular dendritic cells, a key player in autoantibody production. Thank you for your attention.
Thank you, Professor, for the fascinating work and the excellent deliberation. Now, I would like to check if there are any question and answers in the chat box. Okay, there is one question that I can see. With regard to the prevalence of HTLV1, is it rational that we find evidence of HTLV1 in blood level, either antibody or by PCR? This question is by Dr. Mehdi, and he has also commented that HTLV1 is prevalent in Khorasan province.
Pardon?
There is a question from Dr. Mehdi. The question is, with regard to prevalence of HTLV1, is it rational to find evidence of HTLV1 in blood level, either antibody or by PCR?
Blood level. I think usually we detect HTV1 by HTV1 antibody. We don't usually use PCR for HTV1. We usually use anti-HTV antibody to detect HTV1.
Okay. Thank you, Professor.
Can
If
I
I
have
may
a
ask,
question? Yeah.
Okay.
Thank you.
Yes, please go ahead.
Interesting. So, interesting talk. Can I just ask whether in the cohort that you have studied, whether did you measure the anti-NMO level, the aquaporin 4 antibody
Antibody level in
this subset of HTL? Because there are some reports of NMO light syndrome associated with HTLV1. And there are considerations that this might be a spectrum.
Usually, HTLV1 is detected only in the endemic area for Japan or coastal area of Japan, including Hokkaido or Kyushu area. So I think the importance of HTV1 is restricted in endemic area compared to EB virus infection. I'm sorry for my poor English.
Professor, thank you for that answer. If I may ask a question, are there any clinical phenotypes which are more prevalent in HTLV1 associated Sjogren syndrome?
Thank you very much. I think the clinical phenotype is not so different. However, the main difference is the seropositivity of anti-Rho SSA antibody. I think.
Are any neurological or hematological manifestations more common in HTLV-1 associated Sjogren syndrome?
I think there is no difference for the including neurological findings or hematological findings. I think the autoantibody prevalence is only difference.
Thank you so much, Professor, for that session. There are no further question and answer in the chat box. If there are any other questions, if there are no other questions, then I would like to conclude this session. And the next session will be moderated by Professor Sumaira Farman.
Thank you very much.